Welcome to the podcast of Hemisphere, the official journal of the European Hematology Association. Hemisphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hello, everyone, and welcome to a new Hemisphere podcast. My name is Francesca Vinchi, and I'm the head of the Alm Research Laboratory at the New Blood Center. In my role as scientific editor of Hemisphere, today I have the pleasure to interview Alessandra Balduini. Alessandra is Professor of Clinical Chemistry at the University of Pavia in Italy and Research Associate Professor of Biomedical Engineering at Tufts University in Boston, US. Alessandra started a research group in 2007 focused on stem cell and megakaryocyte biology and clinical aspects of diseases related to platelet and clotting processes. Welcome, Alessandra. I hope you are well, and it's a pleasure to have you for this podcast.
Hi, everyone, and thank you, Francesca, for the nice introduction. I'm really happy to be here and to answer your question and see what we can do in the future for megakaryocyte and platelets and bone marrow.
So I would like to kick off this conversation asking, how did your passion for platelets start off? And what was in your initial focus? We all know you are very much interested in platelet biology since many years. So my curiosity was, how did story started?
So I'm a medical doctor, and when I finished medical school, I studied in France and in Italy. I did my postdoc in the States working on stem cells. So I was very interested in studying stem cells. I did my postdoc with Hal Broxmeyer in Indianapolis. Then after I came back from Indianapolis, I was a clinician for a long time doing residency, and then I was the head of the anticoagulant therapy for a long time. And so my interest in platelets started. And then I combined my interest with stem cell on my postdoc with my clinical job. And I went for a sabbatical to Harvard Medical School to study megakaryocyte and how platelets are generated. And then at the end of this sabbatical, I decided that I prefer to do research, and then I quitted my job as a clinician, and I started my own group in 2007. So this is the story, and this is why. So I combined research and my clinical interests to start my lab.
Okay, so next question, and this is something I'm very curious to hear about. So you are effectively reality well known worldwide because of the bone marrow scaffold you have generated starting from silk cocoons for the study of platelet production, so the so-called silk fusion technology. Can you tell us where this idea came from and how it allowed you really to investigate megakaryocyte formation?
Yes, again in Boston, because during my sabbatical, I wanted to. I was working with Joe Italiano and all the group, Ramesh Shivdasani, all the group in Boston, but I wanted to do something for mimicking the human situation. So studying platelet formation in human and patients, and as you know better than me, the bone marrow is a difficult organ because we cannot see it. So I started to think about developing a model. And then I was searching for a material to build this model. And I read a paper where they were describing the use of silk in the filter for dialysis, because silk doesn't activate platelets. And this was known for a long time. So I started to search, and again, serendipity, because I was introduced to David Kaplan, who is the director of the Department of Biomedical Engineering at Tufts, was at the time. And he is the one that discovered silk as a biomaterials, and he didn't know anything about blood or bone marrow because he was using silk to mimic other organs. He's a biologist. And then I asked him if I could use silk and if I could work in his lab and in his department to see if silk was a good material to mimic the bone marrow. And he told me, yes, you can. And if you succeed, you can stay here forever. So I'm there since 2007. So this is the story. And then I got involved in silver materials. And then I started to work in Boston for the materials, and in Italy, in Pavia, for more the clinical application, because my lab is near the hospital, and there was a clinician here. And then the story started like this.
Wow, great stories, and actually congratulations for your great intuition on silk. So how did you further develop the use of this bone marrow scaffold? So basically along the years, what are the discovery brought you, and what would you say it was the most interesting discoveries that your lab made using this particular scaffold technology?
So this year, my first silk bone marrow model turns 18. So in Italy, it's big. It's big. So I've been studying the bone marrow models for 18 years now, and my lab is started 18 years ago. So now we have a pipeline of different silk model, and the advantage of silk is that it doesn't activate platelets, but it's also a very versatile material that you can functionalize with different components of the bone marrow. We started studying bone marrow, studying extracellular matrix components that were protein that were not studied a lot in the bone marrow for bone disease, yes, but not the bone marrow. Also, we started making these sponges of silk, mimicking the structure of the bone marrow with matrices, and then we developed the perfusion of the system to mimic the blood flow, and then we developed the system to mimic the niche also in terms of elasticity. Not only the biochemical composition, but also the structure and the physical properties of the silk. And then the application are many. Now we can produce platelets that they look like peripheral blood platelets, our platelets, and in vitro they are functional. We have also a paper in revision now in producing red cells. So we can also produce red cells. So we can produce different blood cells using the silk. But I think that the most interesting application at the moment is that we can mimic diseases. So we can mimic disease, and we can also do drug testing, and we have a couple of publications, because another publication came out last week on using this silk system. We also have silk bioink to predict the response of patient to drug using patient blood. So we developed system to culture megakaryocyte or blood cells in general for a very small amount of blood, and we can test drugs and see if the patient will respond or not. For example, we used the thrombopoietin receptor agonist, the thrombopag, to see how patient can respond to the drug.
Can I ask you, is there any limitation in terms of amount I can imagine of the platelet production with the silk scaffold? But I was wondering whether for transfusion medicine purposes one day will be possible to have bigger amounts. Do you think this is something, I mean, obviously you use the system for research purposes, but I was wondering whether this technology can be applied in larger scale for transfusion medicine. Would this be something that you see could happen in future?
In the long run, yes, it can happen, but in the long run, and of course, the work of Koji Ito in Japan, he has been the pioneer in this field, sure. And we have to recognize what we did. But from my side, what I can say, and we have a big network also with the people in Boston, in Paris, many people. It's a small field, but we are very connected, because you need two components to produce platelets. So you need a cell line, an IPS for the moment, because it's the reproducible cell that you can use to produce platelets, and then you need an environment. My belief is that if you have a right environment, if you can mimic the physiology of the bone marrow, then you can improve the number of the platelet released. For example, with our new system, the Silk BioInk, where we reproduce the exact elasticity of the human bone marrow, we had a huge improvement in platelet production. That means 5 times more. So it's really we were around 20% of megakaryocyte producing platelets, and now we can have 50% of megakaryocyte, 60%. It's really, and they are more mature and they are producing more platelets. So just mimicking the bone marrow. So you think if you combine the two technology, I think you can have a much better result. And in other words, I think the only solution is combining the two technology, the niche in one side and the cell in the other.
Sure. So you have been involved in hematological research using these 3D scaffolds since now, as you mentioned, a long time. So can you give us your point of view how using these scaffolds has really revolutionized our knowledge in hematology in a more extended manner, also beyond your own discovery?
I think it's really crucial in general, not only for bone marrow, because I think that what the experimental hematology field is doing is mimicking the niche, and in general what the cell biology is doing for different organs is mimicking the structure of the organs to study how they work and to study humans. And I think this is really a revolution. We have organoids now, we have organ on a chip, we have 3D models and so on. I think that for the bone marrow needs a special attention because it's not an organ like others, and also because it's made of different components. It's inside the bone. We don't really have any information about the human because we cannot see it. We just can sample it. So I think this is crucial to understand the composition, the function, and how the different components interact with each other. I have data that are too preliminary to be presented, but I have data that show that it's important to mimic the 3D structure and the physiology of the bone marrow in order to have cultures that are more reliable in terms of replicating the diseases or in general the human function. I think this is a revolution for in all the field that study 3D models.
So in one of your last publications that I also had the pleasure to highlight in Hemasphere with an hematopic, your lab unveiled the involvement of mechanosensation in the regulation of megakaryocyte maturation and proplatelet biogenesis. Can you just give us a brief overview of the findings of this study?
Yeah, so this is a work in collaboration with Katia Ravid in Boston and Catherine Leon in Strasbourg, and it's based on the study my lab has done on mechanoreceptor and in general calcium signaling. So calcium metabolism is very important for megakaryocyte function and platelet formation. So starting from this knowledge, we studied mechanoreceptors that are ion channels and calcium channel in this field. So we published a first paper on TRPV4, that is a mechanosensor that has a positive impact on platelet formation. So its activation leads to platelet formation, while piezo-1, the opposite, is a brake on megakaryocyte maturation and platelet formation. We are studying now why the exact mechanism, because the exact mechanism is not known yet. But what is interesting and what I think is always important for this model is, again, trying to replicate the human phenotype. So we discovered that piezo is a brake on proplatelets and platelet formation, and it supports more megakaryocyte proliferation, so the proliferation of small megakaryocytes. So to confirm this data, we went to the disease, and a disease in the bone marrow where megakaryocytes proliferate and where megakaryocytes are smaller than normal is primary myelofibrosis, where megakaryocytes are the diseased cells, and they proliferate, and when the fibrosis increases, then you have a decrease also in platelet production, and an increase in megakaryocyte proliferation of small megakaryocyte. And this megakaryocyte in primary myelofibrosis, they have an overexpression of piezo-1. So again, this is consistent with our data, because when megakaryocytes are small and they have a defective platelet production, then they express more piezo-1. And the same happened in mouse models. These experiments were performed by Katia Ravid in Boston, and the same happens also in mice where piezo-1 is knocked out. So if you knock out piezo-1, and this experiment were done by Catherine Leon in Strasbourg, then you have an increase in platelet number in the mouse. And what is interesting is that when you inhibit piezo, then you can revert the phenotype also in patients. So megakaryocytes become a little bit larger, and they start producing platelets again.
That's very interesting. So maybe in the future, piezo is a target in myelofibrosis, right? So I have still a couple of questions. One is still more scientific focus. So how are you further developing in your lab the silk fusion technology? Meaning, what are the new direction you are now working? You were mentioning, for example, let's say mimicking the niche, the bone marrow niche for certain hematological diseases, something now that you are focusing. Can you give us some hints about what you are doing on this?
So yes, it's different direction. So for the bioreactor, so the mimicking the niche, I'm focusing on the niche. You know the story: good seed in a good niche, in a good soil, bad seed in a bad. So because in the bone marrow again, it's special because if you cure the stem cells and you don't cure the niche, then you still have the disease, even if the niche is not the one that is start to be altered with the disease. So my goal is trying to mimic the niche in disease and in health, to understand what's going on, and to understand the mechanism of disease also in terms of the niche, and in terms of the relationship between hematopoietic stem cells and the niche. And now is in general, so it's stem cell, and as I told you, we are also studying how red cells are produced, and also platelets. So understanding really the capacity of stem cell to differentiate. And then with the new model on the list, that is this silk bio ink that mimics the elasticity of the bone marrow. I want you to understand how elasticity and the softest of the bone marrow, because the bone marrow is the softest organ we have in our body. It's softer than the brain. So I'm interested in understanding. Yeah, nobody knows that. Also, it's interesting. It's really So I'm interested in understanding how important it is to mimic also this feature, with a particular attention also to drug discovery and drug testing. As I told you, we can predict the response to patient. We can use few sets of patients and still understanding the mechanism. So I think this is a good tool to understand disease and to do drug discovery and drug testing. Doing that, we can understand new mechanisms that can lead to the scale up of platelet production or red cell production in the future, maybe. And I think this is the way to go. So always validate your system with the diseases, and see if you can replicate phenotype of diseases, so you can understand that you are doing something good for your model, because it's mimicking something real.
what can you share something on what diseases are you modeling? You a little bit you work on myelofibrosis, but let's say more in the direction of platelets. Are there some specific disease you are modeling?
So I'm interested in primary myelofibrosis and in myeloproliferative neoplasia in general, and then my first interests are of course inherited thrombocytopenia and inherited diseases of blood. So I'm also studying a lot of different inherited thrombocytopenia, and I'm also back to studying MYH9 related disease that was I published on that long time ago, but now I'm going back studying it. Bernard Soulier, classic disease. I mean, the first inherited thrombocytopenia is diseased, but we still have many mechanisms to discover that we don't know yet.
So as the last question, I have a bit more personal question. So I would like to touch a point when you can bring a little bit about your experience as a scientist. So you are indeed a scientist at the crossroad between two continents. You have worked both in US and in Europe, in Italy. So can you tell something that you love about doing research in US and something you love about doing research instead in Europe? And how experiencing both continents has also influenced and changed your career?
So this is a difficult. It's a difficult question. And speaking between Italians, we know how difficult it is to be in Italy. But I think it's a fundamental question because everybody knows about the big research in the States, while Europe is more difficult. So what I can say, starting from the positive, is that in Europe you have very good clinicians. Medicine is very good in Italy. I work in Pavia, where platelets are discovered with 2 of the 3 mutations of myeloproliferative neoplasm were discovered. The hematology is very strong in Pavia. They had a story of experimental hematology, but not like I'm doing. I'm bringing new things in Pavia, a new approach as experimental hematologist. And the other positive thing is that I think my real motivation to stay here and not to move to Boston for good are the fact that I can have a stable group here, and I have to mention them. So Alessandro Malara, Cristian Di Budo, and Vittoria Bonante. These are the 3 young. They're not as young as before because they started with me, and we started this adventure together. And I can say that I think this is the main reason why I stayed in Italy. The fact that I could have now they are all associate professor. I'm full professor. They are associate professor, but we can be a group, and this is fantastic and very important to carry on the work. The States is my energy. Without the States, I would be depressed because
survivor.
Just I have to be honest. I think it's important also in this podcast to be honest, so we can adjust what's research. So
I do agree with you.
Yeah, I know, because I like your personality.
For a long time in Italy and then in Germany, and now I'm in US, so I understand what you mean from this point of view.
You understand what I mean. It's not a question of amount of funding, because I think that if you have the energy and if you are interested, you can do whatever you want, also with small funding. And in Italy, in Europe, it's not true that we have small funding. Now I have 3 European projects. It's a lot of fundings, and I can really do many experiments. I have a modern lab. Nothing to complain about the structure, but it is the energy, and also is the fact that people don't support you as in the States if you work a lot. In the States, if you work a lot, they like you, they support you, they smile at you, they love you. Here, no, and this is my difficulty.
I do understand. Very well pointed out. Yes, I understand. And the energy and the vibe you get from US from doing this job, it's really strong and it's empowering. And I think also for a woman in science, especially,
exactly.
It's very important. So thanks for pointing this out. So, Alessandra, it was a pleasure to have you for this podcast. I hope you enjoyed this last Hemisphere podcast, which is now over. Thanks again, Alessandra, and thanks everyone for listening.
Thank you, Francesca, and thank you, everyone. Thank you so much.
Thank you for listening to this episode of the Hemisphere podcast. All of Hemisphere's content is open access and can be found at www.hemispherejournal.com. We hope you will join us for future podcast episodes.